Characteristics of alcohol metabolism in the liver from a pathophysiological perspective. Consuming alcohol in large quantities or on a regular basis can lead to alcoholic liver disease, ALD, which is a chronic liver disease that is prevalent all over the world. Before starting the video, please do not forget to like, share, subscribe, and press the bell icon for new interesting videos. For this reason, the liver is especially vulnerable to the effects of alcohol consumption because it is the primary organ responsible for the metabolism of alcohol. A number of mechanisms, including the impairment of lipid metabolism, the intensification of inflammatory reactions, and the induction of fibrosis, are responsible for the damage that is caused to the liver by the metabolites and byproducts that are produced during the degradation of alcohol. The absence of animal models that are capable of accurately representing human ALD has been a barrier to the development of innovative treatments, notwithstanding the severity of ALD. For the purpose of overcoming the constraints that now exist in ALD investigations and the development of therapies, it is vital to have an understanding of the molecular pathways that are responsible for alcohol-induced liver impairment. The purpose of this study is to provide insights into the course of alcohol-related liver disease, ALD, by analyzing past studies that were conducted on alcohol liver metabolism. In particular, there is an emphasis placed on the incidence of ALD that is brought about by hepatotoxicity that originates from the metabolism of alcohol. Alcohol has been integrally associated with human civilization for a very long time, and it has also been used as a drug and regarded an indulgence. The intake of alcoholic beverages is a substantial contributor to the significant increase in morbidity and death that is occurring all across the world. The trend of alcohol intake is constantly increasing, despite the fact that there is a growing awareness among the general public about the hepatotoxicity induced by alcohol. The consumption of alcohol is known to hasten the progression of liver damage in individuals who are infected with the hepatitis virus. Cirrhosis caused by alcohol is responsible for nearly half of all deaths that are associated with liver cirrhosis. Abstinence from alcohol is the most essential method for preventing and/or reversing the advancement of ALD. Nonetheless, it is difficult to suppress the recurrence of drinking because the majority of patients who have ALD are alcoholics. Similarly to other end-stage liver disorders, a liver transplant is regarded to be the definitive treatment for alcoholic liver disease, ALD. On the other hand, liver transplantation is a challenging therapeutic option because it is dependent on the availability of donors and requires abstinence from alcohol for a minimum of 6 months before, to, and following the transplant. Additionally, there is a concern from an ethical standpoint that the transplanted liver would be wasted on a patient who subsequently returns to drinking, which would cause the transplanted liver to become damaged. Furthermore, there is no medicine that has been approved by the Food and Medicine Administration, FDA, that is highly effective in treating patients who have ALD. Consequently, it is essential to discover effective medications for the treatment of ALD diseases. In order to develop treatment methods for alcohol-related liver disease, ALD, it may be helpful to get an understanding of the process of alcohol metabolism in the liver as well as the effects of hepatotoxic intermediates implicated in the progression of ALD. This article provides a summary of the metabolic processes of alcohol as well as a discussion of the pathogenesis of alcohol-related liver disease, ALD, which provides insight into the underlying mechanism of ALD. 2. The metabolism of alcohol. Consumption of alcohol results in its absorption through the intestines and the stomach. The amount of alcohol that is expelled through the breath, perspiration, and urine is less than 10%. It may be deduced from this that more than 90% of the alcohol that is absorbed is distributed throughout the body and is ultimately carried to the liver through the portal vein. Because it contains a significant amount of enzymes that are responsible for the metabolism of alcohol, the liver is an essential organ in the process of alcohol metabolism. There are 2 stages that make up the oxidative route, which is the primary mechanism for the metabolism of alcohol compounds. To begin, alcohol is converted into acetaldehyde by the action of alcohol dehydrogenase, ADH, which is the major enzyme responsible for the conversion of alcohol to acetaldehyde. Oxygen deprivation leads to an increase in the expression and activity of cytochrome P450-2E1-CYP2E1 rather than adenosine dehydrogenase, ADH. Activated CYP2E1 is responsible for the creation of reactive oxygen species, ROS, which in turn increases the production of acetaldehyde. In addition, peroxisomal catalase is responsible for the conversion of alcohol to acetaldehyde. However, due to the fact that it plays a relatively modest role in the process of alcohol digestion, its action is regarded to be a minor pathway. Aldehyde dehydrogenase, ALDH, is responsible for the quick conversion of acetaldehyde to acetate, which is the second step in the oxidative process. It is not the liver that is responsible for the conversion of acetate into carbon dioxide, CO2, fatty acids, FAs, and water, H2O, but rather the peripheral tissues. In terms of quantitative analysis, the non-oxidative route is responsible for a relatively insignificant percentage of the alcohol metabolism. Different enzymes are responsible for the non-oxidative conjugation of a modest amount of alcohol to a variety of endogenous metabolites of varying amounts. For instance, the formation of fatty acid ethyl ester (FAE) via enzymatic esterification of alcohol with FAs and the formation of phosphatidylethanol (PEth) by transphosphatidylation of phosphatidylcholine with ethanol are both examples of processes that are catalyzed by phospholipase D (PLD). Furthermore, the production of ethyl glucuronide (ETG) and ethyl sulfate (ETS) is a consequence of the conjugation of alcohol to glucuronic acid and sulfate, respectively. One of the driving forces behind the progression of alcohol-related liver disease, ALD, from alcoholic steatosis to alcoholic cirrhosis is the substances that are produced during the process of alcohol metabolism. Acetaldehyde is the most well-known poisonous chemical that is formed as a byproduct of the metabolism of alcohol. A direct interaction between acetaldehyde and DNA results in the formation of point mutations as well as damage to chromosomes. In addition to this, it forms acetaldehyde adducts by binding to a wide range of proteins, which causes the structure and function of the liver to be altered. 3. Oxidative ethanol metabolites play a role in the pathogenesis of alcoholic liver disease. In the liver, oxidative ethanol-derived metabolites are responsible for a wide variety of harm, including fat buildup in hepatocytes, inflammation, fibrosis, and the development of cancer. An excessive accumulation of lipids in hepatocytes leads to the death of a large number of hepatocytes, which in turn causes responses that are pro-inflammatory and pro-fibrogenic, hence raising the likelihood of developing liver cancer. In light of the fact that there is a paucity of evidence regarding the pathophysiology of ALD that is driven by non-oxidative ethanol metabolism, this section will discuss the processes that are responsible for oxidative ethanol metabolite-mediated hepatotoxicity that is initiated by excessive alcohol consumption. Excessive accumulation of lipids in hepatocytes has been seen. When there is an excess of lipids and the mechanism that removes lipids is compromised, hepatocytes tend to accumulate lipids. A process known as de novo lipogenesis is responsible for the former, which takes place when the liver increases its uptake of exogenous FAs or lipid synthesis in the liver. Acetaldehyde, in particular, is recognized for its ability to stimulate lipolysis in adipose tissue and to increase the quantity of free fatty acids, FAFAs, that are absorbed by the liver. This process is facilitated by the hepatic FA transporters, which include FA transporter proteins, FATPs, and FA translocase, fat CD36. Furthermore, it has been discovered that the overexpression of fat TP2, fat P5, and fat CD36 has been observed in liver tissues of rodent models of alcoholic liver disease, ALD. Additionally, acetaldehyde is responsible for the dysregulation of the 5-adenosine monophosphate activated protein kinase AMPK pathway. This pathway is responsible for the regulation of the expression of lipogenic transcription factors, such as sterol regulatory element binding protein 1C, SREBP1C, and carbohydrate responsive element binding protein, TREB. Consequently, there is an increase in the expression of genes that are responsible for the production of lipogenic enzymes such as acetyl-CoA carboxylase 1-ACC1, fatty acid synthase FASN, and steryl-CoA desaturase 1-SCD5. On the subject of lipid homeostasis, lipin-1 is involved. In the nucleus, hepatic lipin-1 either performs the function of a transcriptional coactivator, lipin, or operates as a Mg2 plus dependent phosphatidate phosphatase, lipin-1b, thereby facilitating the production of triglycerides and phospholipids in the cytoplasm. Mitochondrial B-oxidation and the secretion of excessive stored triglycerides that are packed into very low-density lipoprotein, VLDL, are the mechanisms that facilitate the clearance of lipids in the liver. It is important to emphasize that the most major factor contributing to the buildup of lipids in the liver is the dysregulation of mitochondrial B oxidation that is generated by alcohol. A factor that splices the RNA splicing factor known as arginine-serine-rich 10, SFRS10, plays a role in the skipping of the lipin-1 exon, which ultimately results in the generation of the lipin-1 variant. Multiple aspects of hepatic lipid metabolism are dysregulated by the hazardous metabolites that are generated by ethanol. These metabolites increase hepatic fatty acid intake and de novo lipid synthesis, while simultaneously decreasing fatty acid oxidation and lipid export. The buildup of hepatocellular lipids is the result of the impacts coming together. Because hepatic lipid buildup is the first indicator of advanced liver disease, ALD, more research on lipid metabolism in hepatocytes may open the door to the possibility of early therapeutic intervention for those who are at risk of developing advanced Alzheimer's disease. Immune system that is not working properly. A growing body of research suggests that drinking alcohol on a regular basis causes disruptions in the intestinal barrier as well as the tight and adherent junctions in the colonic mucosa. This, in turn, facilitates the movement of lipopolysaccharide, LPS, into the circulation. The adverse effects of acetaldehyde contribute to this by increasing the expression of microRNA 212 in enterocytes and decreasing the expression of zonula occludens 1, ZO1, which is a component of tight junctions. Kupffer cells, which are macrophages that reside in the liver, are activated by both acetaldehyde and LPS. This activation causes the generation of reactive oxygen species, ROS, and chemokines, which in turn attract bone marrow-derived neutrophils and blood-derived monocytes into the liver. ALD is referred to as alcoholic hepatitis when it reaches this stage.